anterior-posterior axes, and a series of genes that become active in the embryo to divide this axis up into territory and tell sort of each group of cells what they need to become in terms of downstream cell fate. Here's a nice quote that I think is really applicable to kind of the principles of today's lecture. It was not written with biology in mind at all, but it really does seem to apply to the chocolate embryo. Okay, so here's our slide that we left off with last time. And remember that we have both maternal components made by the mother, made by those nurse cells within the egg chambers that have been shifted over and placed in the Drosophila oocyte. And then we're also going to have a number of different segmentation genes that are all transcribed from the zygotic nuclei within the embryo itself. So last time we were talking about bicoid as being an important morphogen in this process, the anterior morphogen. And so by this concept, you take a single gene product and you just vary the concentration. So this is arranged in a gradient across the embryo, highest to the anterior and tapering off as we get more posterior. And just based on this information, we already can start out the process of dividing that anterior-posterior axes into different regions. So this is a gradient. We talked about some other gradients with nanos at the posterior. And I think it's fairly intuitive to think about how that works. So the first big question for today is how do we get from a gradient like this to a downstream expression of targets of bicoid and nanos and the other transcription factors, where it's a very chunked out type of expression. So this is showing you a Drosophila embryo. It's in a immunohistochemistry. And each different color reflects an antibody that has been made against either hunchback, cripple, or giant. And these are 3 of the gap genes expressed by the embryo. And you can see the punctate nature of it. This, of course, is because all of these factors are transcription factors. So you're really picking up staining within the nuclei of this embryo. The other major thing that you'll notice is the fact that these are sort of wide bands, maybe 5, 10, 15 cells wide. But then on the edges, they really have a fairly distinct border. So crupal here in the middle, very high expression just within this domain, and then only a couple of border cells are sort of medium, and then that's it. So how do we get from a gradient to blocks of expression? And this is going to involve regulation by maternal protein products and by other gap genes themselves. So almost all of the factors that we're going to talk about on that diagram that was previously shown on the previous slide here, almost all of these things are transcription factors. So picture proteins in the cell that are landing on target recognition sites within the promoters, within the enhancers. And when those transcription factors land on these target genes, they will appropriately activate or repress the transcription. Okay, so this sort of gets to that same idea, is that the maternal gene products, bicoid and nanos, both of these are transcription factors. They're going to be actually regulating the expression of these gap genes. And then once the gap gene proteins themselves get made, they'll come back immediately and start cross-regulating each other. So the gap genes are so named because of their expression pattern in kind of wide bands, and their very early expression in the embryo. You had a question?
Yeah, the original maternal genes factors, those gradients, those are protein gradients, transcription factors. Was that started from MRI gradients, or did, I'm sorry, you might already mention this, or did the MRIs get transcribed into proteins made degrading themselves?
Yeah, good question. Are we talking RNA gradients or protein gradients here from these maternal products? And both. So the maternal RNAs tend to be, if you assay by in situ, they look like they're a little bit more squished up to one side, anterior or posterior. but definitely by the time you get to proteins, they're in a very nice gradient across the anterior posterior. So,
Those are microtubules, I think I start remembering now. The
Sorry?
microtubules you said that kind of pull the proteins.
right, so
up.
microtubules in the oocytes, that was one of the mechanisms, one of the actual how do you get this done type stories for bringing these specific mRNAs either to anterior or posterior sites. Any other thoughts or questions at this point? Okay, so we're going to see cross-regulation, and this theme will continue as we go on down talking about the other classes like the parallel genes and the segment polarity genes. So there's a certain order in terms of timing that genes are going to be made, gap genes first, and then parallel genes and the segment polarity genes. And part of the reason for that timing is that you need some of these earlier proteins to be made as transcription factors and turn on some of the downstream genes in these different classes. Okay. All right. Okay, so we have bicoid. It's a morphogen. It's in a gradient. So we could draw a little diagram that looks like this with bicoid concentration on one axis and the larval anterior-posterior distance along the bottom axis. And so maternal bicoid protein is graphed there, and what we will see is that a particular target gene, gap gene number 1, needs a certain minimal level of bicoid in order to turn on. So if we ask where on this axis is that condition met, well, it's met basically from about the center point and more anterior to that. These are the regions, this is the territory within the embryo that would have at least that much bicoid present in the nuclei of the cells. So, if that much bicoid is present, these gene products are able to be activated, and that defines a certain window of expression for gap gene number 1. And perhaps gap gene number 1 is something that you need to build the apron type of tissue or cell fate. So remember, apron was the most anterior types of structures that we would see in the larvae. Okay? So that is gap gene number 1. It has its own sort of threshold level. We could talk about a second gap gene, number 2, which also regulated by bicoid, and might have a somewhat different threshold for responsiveness to bicoid to be turned on. So this gap gene number 2, it's a little bit less picky. If we have a slightly lower threshold, that's still fine. So anywhere from that point on and up, now you're going to get gap gene number 2 turned on. Gap gene number 3 perhaps does not respond to bicoid, or may even be negatively regulated by bicoid. So it would actually want to be expressed perhaps in an area where there's little bicoid available. Okay, so then in addition, now we have an area that has 2 gap genes together, which is a somewhat different scenario than one gap gene by itself. So we're going to get different combinations of transcription factors being activated in these cells. So that's a way to make those cells different within the embryo. And it may be a simple example here that in the region where you find gap gene 2 only turned on, that's a different information than gap gene 2 plus 1, so we can say head will be specified in this territory, apron will be specified in that territory. So there are some nice little animations and videos and a nice tutorial in this website called FlyMove. And I meant to tell you, I made a new folder on RAMCT that has websites and videos. So a lot of the videos that I showed in the class, or maybe you didn't have time to show, if you want to go and look at those on your own, you can find them there in that folder. So look for the website called FlyMove, and there's a lot in the section called segmentation genes, including a number of these different animations, which I think are kind of fun to play with, but also sometimes seeing a picture or a little animation, it just gives you the intuitive concept right away. Okay, so this is a wild type situation, and in the wild type situation for bicoid, we have a certain proper amount of bicoid that can make a nice gradient going from anterior to posterior. What we'd really like to do next to sort of test our hypothesis regarding this sort of show it or correlative information is to perturb that condition. So we need to look at some mutants. So a really informative mutant to look at in this case is actually a situation where you might make only half as much bicoid protein as normal. Okay, we could also just eliminate bicoid protein, but we already know a little bit about what that phenotype is. It shows us that bicoid is needed to make anterior sulfates, because remember that larvae just lacked all anteriorness if we got rid of bicoid. So here's a situation now where we have a mutant, maybe a heterozygote mutant, or maybe a mutant that has some partial function left within the mutant bicoid protein. But suffice it to say that now in this condition, only half as much bicoid protein could be made in the embryo at its best. So over here in the anterior, most cells, they're doing their best to make bicoid, but they only make half as much as wild type. And then we get a gradient that's spread out like before, but it's not quite as high in terms of the amount of bicoid present in these cells. So the question is, what will be the response of the target genes, and where will those target genes be activated? Now the target genes are not changed in our mutant. They're same old wild type sequence just like normal. And they're going to respond to bicoid just like normal. So we said that gap gene number 1 in green, it needs a certain threshold, like 50% of maximal, to turn on. Well now, the only cells that meet that condition in this mutant scenario is a small web of cells that are very close to the anterior end. Does that make sense? Yeah. So that's where gap gene number 1 is going to turn on, in now only a very small slice of cells. Gap gene number 2, again, it has its unique and unchanged response to bicoid, but now in this mutant scenario, there are fewer cells that meet that condition. So you get a smaller band of expression for gap gene number 2. On the other hand, gap gene number 3, it prefers low bicoid. So it may actually have a broader region of expression for that gene. Okay. So you can see how tweaking the morphogen level, you just automatically tweak the expression of all the downstream target genes as well. So now, this will give us a very narrow group of cells for making acron, gap gene 1 plus gap gene 2. So we may expect the acron structures maybe to be present, but in a very minimal amount. Whereas the posterior structures, so let's say we have a larvae here, the posterior structures may be quite a bit expanded in this particular mutant. So we've just, almost like a sliding scale, moved the patterning response of this mutant embryo. Okay, here's a nice animation. You can look for that icon in the fly move tutorial and you can play with that yourself. Okay, so taking it one step further, here's a little bit more sophisticated diagram looking at several different gap genes. Hunchback, Krupel, and Spherical talus. You don't need to know these names. And in addition to having a, say, minimal threshold for turning on, some of these genes also may have a maximal threshold, where you need a certain amount of bicoid, but not too much. So they're very particular about exactly how much they will respond to, and too much is bad, too little is bad. And if you start to put together multiple different gap genes, you can really get a very fine level of differentiation, that is a fine level of differentness across this anterior-posterior axis. And this will help that embryo to divide up the anterior-posterior axes into different regions or territories that could become then the appropriate types of structures. So just to complete the thought here, B is a gradient, such as bicoid, that may be very high, medium low, or low in the embryo. That could be an mRNA gradient. C could be a protein gradient for bicoid. And then D shows you the expression of the gap genes, which are more in a chunked out type of expression pattern. Okay? Isn't that neat? It's just so elegant, I think. You know, we just go from gradient to these chunked out domains of expression. Any questions? Okay. So in fact, we don't just have Bitcoin to worry about. We have 4 different maternal gradients that are found in this embryo. These are all maternal products. Hunchback is sort of a dual situation where there is some maternal hunchback provided, but it's also activated from the zygotic genome once we get to that point. So it's sort of both the maternal product and the zygotic product. And so these 4 gradients are all transcription factors, and they're all doing the same sort of thing as I just told you about for Bicoid. And so this gives us a lot of fine maneuverability to turn on gap genes within these sharp expression domains. Okay, so each location on the anterior-posterior axis having a unique combination of maternal products and therefore gap gene expression. Yeah?
So you were saying that the gap genes are the zygotic genome starting to transcribe. So that's what is the mid-blastula transition is when that's starting to happen.
Correct. Yes.
So with the vertebrates, that it happens real fast. All this happens like
For vertebrates?
yeah. Because you're saying the mid-blastula transition for vertebrates happens like almost immediately, where these other ones are drawn way out.
Yeah,
So
absolutely.
is all this happening real quickly with
So,
them, or is this a different story with vertebrates?
It's an excellent question, and it's a different story. So, basically with Drosophila here, because of these screens done by Nisla and Bolhard, and because we know so many of the key genes, we've been able to map out the mechanism of how this happens in the Drosophila. So it gives us a concept of how this could be done. But, you know, different organisms are going to do this in a different fashion, and vertebrates don't really rely on maternal products in this way, and they don't have a similar paradigm for establishing the anterior-posterior axis. So they do it their own separate way. All right. This is a little analogy that I think can be useful. So what we have here is a cute kid, and he lives in a neighborhood in the purple house, and he has, he likes to go out and play in this neighborhood, but there are some constraints here. There's a dangerous train track on one side, there's a dangerous dog on the other side. So initially the parents might have told him that it was okay to go out and play as long as he stayed within the neighborhood. Okay? But then Big Sister told him, you know, be sure to stay away from the train tracks, and his older brother told him, be sure to stay away from that angry dog. So if he listens to all of his siblings and parents, he will then only go out to play within this very restricted region. So they're each giving a distinct input, and combining all these together defines his window of flexibility. Okay, so the parents could be analogous to Bicoid and Nanos, because those are the maternal products that are placed in here. The brother and the sister, that could be analogous to gap genes, because we're within the same class here, but they're cross-regulating each other. So when you look at a gene now, you can think about the same sort of thing going on, where if we look at Krupel, one of the gap genes, and how it's expressed in the embryo, it needs to respond appropriately to the positive or negative regulation of any of the maternal products and any of the gap genes that are there. So in the case of Bicoid, it activates Krupel, so that normally would make a very long window for Krupel to be expressed in the anterior portion of the embryo. But on the other hand, we have Hunchback is repressing Krupel. So that's going to eliminate some of this highly anterior portion of the embryo, because that's where Hunchback's highly expressed. And so to meet both of those conditions, now Krupel has to be expressed in only a middle portion of the embryo. Nervus is down toward the posterior end, and it's limiting the border of expression from that side of the embryo. So that's going to give you a broad swath of expression for the gap genes. If we come back to our analogy, and let's say that there was a tsunami that came through and it destroyed this train track. Well, now, we still have the constraint of playing in the neighborhood, we still have the constraint of the angry dog, but we don't really need to worry about not going to this territory because it's no longer dangerous, there's no more trains going by there. So in this mutant condition, now we can change or expand the region where the kid is safely allowed to play. And if we look then at one of the regulators of Krupel, if we have now a mutant for hunchback, we've lost one of the steps of regulation here. We've lost the prohibition for Krupel being repressed in the extreme anterior. And so now the Krupel expression can expand to take up all of this territory of the embryo because it's still meeting all the conditions imposed on it by the genes that regulate it. Okay, so in this little animation here, what you're able to do is to click on one or another of these genes that are expressed in different types of territories and are cross-regulating each other and see how the response of a target gene will change in its expression pattern based on altered regulatory transcription factors that are either present or not present. Okay. All right, so those previous 2 or 3 slides, it's sort of a concept of explaining how does one gap gene come to be expressed within a narrow territory of cells. So that might be a good study question for you to think about. Could I actually articulate this, explain this to my roommate? And if so, then you've got the concept. Okay, so why would we call these things gap genes? And in fact, the reason for that name comes from the mutant phenotype that arises when the gap genes are missing. So in this Krupel, we said tends to be expressed kind of in the middle of the embryo as it's forming. And the embryo is going through this transition here from syncytial blastoderm to cellular blastoderm to the status where we have morphologically recognizable segments, to then a little bit later in time where we have a larvae that has morphologically recognizable features such as small little bristles or clear areas within each of these morphological segments. So in purple here, this is showing you where Krupel would be expressed in the wild-type embryo. If you look at a Krupel mutant, it looks like this. It's a short, shrimpy larvae. But in fact, if you look in more detail, you can see why it is a short, shrimpy larvae. And that is because it's actually missing all of these segments that have purple here. So those are the segments that were mutated, or segments that were not able to express Krupel, and those would be missing in the embryo larvae once it develops. So this creates a gap. We still have some acron tissues right at the very top there, and maybe segment number 1. But then we're missing a bunch of segments, and then we pick up with the reappearance of the most posterior segments. Okay, so there's a gap in the body plan, and genes that can mutate to produce this phenotype are called the gap genes. Okay, here's a couple of examples. So we see here some different mutants. First one is Krupel, and these have been stained to reveal those white spots are the small denticles or the small little bristles on these larvae. And here we have the 3 mutant larvae, which are all shorter than normal along the anterior posterior axes. But looking in more detail, you'll see that each of these is a unique phenotype. So this Krupel here, it's missing many of the anterior post segments. A1 through A5 are gone. This middle guy, A1 through 5, are present, but we're missing a couple of the thoracic segments. And then that guy over there at the far side is missing a different combination of segments. So each gap gene, is expressed in a different broad swath of territories, and mutating each gap gene will give you a unique larvae with a body pattern disrupted based on where that gene would have been expressed in the wild type. Okay. So the gap genes are beginning to be expressed in the syncytial blastoderm as the embryo is transitioning to the cellular blastoderm. And now I want to look at the next group of genes called the parallel genes. And these are going to be expressed in stripes, and we have, let's see, 1, 2, 3, 4, 5, 6, 7 stripes here. And, you know, I don't know if you guys are used to seeing, you know, regions of expression within embryos, probably you're not, but if you were, you would know that it's very unusual to see genes expressed in stripes. You know, why? Why would they do that? So, when this was first found, it, you know, raised a lot of interest because of that unusual nature to it. So, before I tell you anything more about parallel genes, I wanted to take a step back and think about what's happening with the embryo at this stage as it's developing these morphologically recognizable regions. So once we get past the cellularization stage, the embryo is going to develop 15 segments. And we can define a segment as being a morphologically discrete region within this larvae that's very, you know, easy to recognize based on visual means. So if you look up there and you counted how many of those chunks there were, you find out there's 15. And so, you know, these were given different names. We're calling them some of the head segments here, teeth or thoracic segments, and then 8 or 9 abdominal segments. And next then, we started to look at genes that were expressed in the embryo at this time, specifically this group of genes expressed in stripes. But, you know, wouldn't you know it, if they're expressed in stripes, the stripes do not align with where the segments are expressed. So here's Futsi-Taratsu. It's one of the parallel genes. And these black bits here are showing you exactly where the expression comes for the Futsi-Taratsu gene. And it's in every other type of segment, but if you look at how this corresponds, it's actually being expressed in the posterior half of one segment plus the anterior half of the following segment. So we still get 7 stripes, but they're not exactly aligned with where the physical demarcations are on this embryo. And so we thought it'd be kind of handy to have a word to describe where in the embryo parallel genes are expressed. And so this offset expression pattern is called the parasegment. And it's exactly the same size as a segment, but it's 50% offset. So does that make sense what the difference between parasegment and segment is? So it's just a different way of how we want to, you know, divide this up and give it a name. Okay. So the parallel genes. This shows you an actual embryo in blue that might represent where one of the parallel genes, Futsi-Taratsu, is being expressed. And these are making the 7 stripes. And the other thing to reemphasize here is that it's not in every single parasegment, it's every other parasegment. Okay, so that'll give you 7 out of 14 or 7 out of 15 parasegments. If you look by in situ hybridization or immunohistochemistry, we can see this in developing embryos. So here are 2 different parallel genes, and they are even-skipped and Futsi-Taratsu. We've made a primary antibody to each of them, and we've actually done a nice dual in situ hybridization or dual immunohistochemistry, where we can recognize now 2 different gene products. And each one of these is expressed in 7 stripes, and in some cases the parallel genes are expressed in all the odd parasegments, and in other cases parallel genes are expressed in the even parasegments. So we just picked one of each and done this dual expression. And now again we get expression in the nuclei, but the width of these regions is much, much narrower than the gap genes. Gap genes was 5, 10, 15 cells wide, and now we're talking maybe, I don't know, 3 or 4 cells wide for these expression patterns. Okay, so just a contrast here between gap genes being expressed in these broad width parallel protein in a much narrower region. Okay, so what about if we had a parallel mutant. What might that look like? And again, we're going to get a larvae that is smaller on the anterior-posterior axes, but this patterning within this smaller larvae is quite different than what we would see for a gap gene. So what has happened here is that the regions where Futsi Taratsu is expressed in every other parasegment, those territories are now just missing. It's as if they were never created. And then everything that was in between is now condensed together, and you get an embryo with literally too few parasegments, which is actually what Futsi Taratsu means in Japanese. So too few parasegments. You could also say too few segments, but probably more accurate to say too few parasegments. So here is just a diagram of what that would look like. And again, we see Futsi Taratsu in every other parasegment, and then those blue portions of the larvae here are deleted, and we join up everything that remains. We get a shorter larvae that has a very specific pattern along its anterior-posterior axes. So we've termed these the pair rule, and I think they decided to use that terminology because of the every other parasegment type of expression for these particular genes. Okay, so one of the things that you can observe with these stripes is a certain crispiness that comes with time. So initially, Futsi Taratsu is kind of expressed in a diffuse way, and then as time goes by, let's say you did immunohistochemistry in a staged fashion over these different embryos, you would see that the longer you let the embryos develop, the more sharp and clear the boundaries were for the expression of Futsi Taratsu. And my question for you guys is, why do you think that happens? Is there a possible explanation that might, yeah?
Well, it's since it's having higher concentration in those certain areas, or it's triggering, it's a transfer two factor. So it's turning those on, how it's going to cascade that amplifies it in one area.